flame. Then place your wire gauge. Wire gauge. Now, the whitish part will be heated until it turns red. Yes.
I have a question.
Yes.
Why does the white steel not burn?
Ni kama tu like mfano mwingine. I'll give you an example which I remember. So now, as you can see, it is evenly heated. So now, it helps in even distribution of heat and also prevents direct heat. Now, what's direct heat? It means that to get clear our beaker on this, it means only one place will be heated. So, on one place. But if you use the wire gauge, the whitish part will help to distribute heat evenly. So our beaker will be heated all the way down. Together. Now let's put our beaker. Then, as we heat 2 leaves. Does our leaf has the requirements for photosynthesis? Yeah? What can you see? Now look at the face.
What are you
The
saying?
requirements. All the necessities for photosynthesis. Does our leaf have those qualities? What can you see in our leaf to make you know that this leaf was able to photosynthesize? Good.
What are you
Chlorophyll.
saying now?
You see now, green color, chlorophyll. Together, so now we did our leaf. Tuangalie ingine ya.
Remember, we are live.
Yeah.
We are live.
Oh.
So they just continue.
So now we'll boil our leaves. Sour. We'll boil them until they boil. Now after boiling, we remove our leaves. Then we boil them again in meditative spirit, but not into direct flame, because you guys are very highly flammable. Hiyo ukishikisha hapo kwa moto itachomeka ujishangae. So we will heat our leaves in a water bath. What is a water bath? We take our leaves. We take, for example, in this our gas jar, then inside the gas jar we put exotic spirits and our leaves there. Then we burn inside this water. Together? Yes. I'm going to tell you the purposes of doing all those processes. Sour. Then from there now, we will wash our leaves using warm water. Warm water. After washing using warm water, we place them on a white tile or a white. We can use those the components for closing our microscope. We place them there. Then afterwards, we pour on them. This is iodine solution. So now we are expecting this color of iodine solution to change from brown when you pour it on that leaf into which color? Oh, this is now brown yellow. Sour. This is now brown yellow. So we expect the color of the iodine to change from the brown yellow into which color? Into blue black. Yes, blue black. Sour. Now, when it changes to blue black, we'll conclude that our leaves photosynthesis, or our leaves made their own food. Tumeelewana mpaka hapo? Yes. Tumeelewana mpaka hapo? Together? So our iodine solution, it will turn from yellow brown or brown yellow to blue black if our leaves had photosynthesized. Are we together? Yes. Can you write something little? Or that? be observing on what's happening on those experiments. You can see on the vision, you cannot see the patches of colorless color. It is almost like turning purple. So, be observing. Again, when you check on the inside of our potatoes, you will see our sugar is full of water. You can just come and observe. Sukari yetu imejaa maji.
Have you seen? Yeah, it's very important to see.
Yeah, that's why we are here for practicals. We are supposed to see. Hata usiangalia kwa haraka.
Unajua mtu anakuja anaangalia.
Ndio vizuri tu.
Come and see.
I've not seen.
Usikuwe na haraka. what we are going to do. So we are boiling the leaf to kill the leaf cells and also stop any chemical reaction. Chemical reaction here is photosynthesis. So you have to boil the leaf to kill the cells. When the cells are killed, it means they won't photosynthesize. Together, it means the leaf cells, they won't photosynthesize. Again, now after boiling, you can see our leaves are boiling. Leaves are boiling. So we want to take them and now boil them again in the methylated spirit. What do we do so? We want to remove the cal or the chlorophyll. Together? So I'm not put this gas here. Maybe it will help us. This is our spirit. Now let me take my leaves. It's really hot. I want water. Oh, I like. Yeah. So now we put our leaves. Remember, while we boiling the leaves inside the water bath using the other spirit, we have said our spirit is highly highly what? So we are saying why we boil our leaf in melted spirit on our water bath because our spirit is highly flammable. It can easily be burnt or it can break into flames. Together? Yeah. Okay. We are waiting for our leaves to boil. Fully boil. say we are removing all the chlorophyll. We want to discolor the leaf. We want to discolor the leaf. Our water has turned to leaf.
Yes.
So this means that our leaves have removed a lot of chlorophyll. Our leaves have removed a lot of chlorophyll. So from there, the next step, what do? We wash it in hot water. There's some water here. We give us some means to be hot. So now check. Our leaves have turned the color even by removing them. You can easily see that. The matte color of the leaf. Remember it was green as this one. So this green. So now see its color. We have discolored it together. Now after here, we want to wash our leaves in hot water to soften our leaf. Remember after using the necessary spirit to discolor it, it will be hard. It will be hard. So we want to wash our leaves in hot water to soften the leaf. Then after that, we'll use those covering for microscope as our white tile and observe whether our leaves had made their food together. So kila kitu kichukua moja moja, yeye hawezi kukatakata. So mchukue moja weke ndio. So we want to use this place as our white tile. Allow us now to wash our leaves. So again, by washing using hot water, it will remove the excess traces of the leather spirit. So the excess traces will be removed by the hot water. The excess traces of the leather spirit will be removed by the hot water. After, we spread it on that white tile. Then now we can pour our iodine. Now we are saying in the presence of starch, our brownish yellow iodine we change from brown yellowish into blue black. Now let's pour. This local some good sincere. It's empty look. Just observe what happens. Just observe what happens. Unafaa kuwa umeanza kuona some traces of blue black. Some patches of blue black. Unafaa kushaanza kuona. Give some time.
This one they are. Yeah, the badges are there.
Unafaa kushaanza kuona patches of
Shaanza kuonekana hapa.
Give it some time. Hizi kwa sawa kwanza kabisa. Black color imeanza kutokea vizuri. How can you full time to blue black? Yote. Kana vile yao inatana vizuri.
Yeah, this one is turning very well.
Yes.
Very well. He is more accurate.
So what do we conclude? And ukienda kutoka tena kabisa. Pende tu ikatana kabisa. Now again, on the same, back to leaf, those are two leaves from the same plant. So, from the same plant. So, two leaves from the same plant. But then you can see one of it, it has much starch than the other one. Yeah? So, one of the leaf, as you compare the change to this other leaf, that one means it has a lot of starch than this leaf.
Even this one has already started.
Yeah.
Badges. Yeah.
So, what can you conclude? Now, I want to go back to the leaf pattern. Leaf pattern. Remember that the way leaves are exposed to sunlight is the way the more they are going to photosynthesize. So, maybe at some point that leaf was more exposed to sunlight than this leaf. That's why you can find that there are high traces of starch on that leaf as compared to this one. So maybe that leaf, that one was more exposed to sunlight than the leaf of the boys. Boys, your leaf was not much. Maybe, we just make a conclusion as to what we are seeing now. So the leaf of the girls, it was more exposed to sunlight than the leaf of the boys. Together? So that's what will happen. So if you don't see the brownish color of adding changing into blue-black, know that that leaf did not photosynthesize, did not make it its food. So there's no presence of starch. What is starch? Starch is glucose. But the byproduct of photosynthesis, we say this glucose and oxygen. So now when glucose is broken down, it is broken down in the form of starch. It is stored in the form of starch in a leaf when it is stored. So now that's why we have presence of starch more in that leaf than in this leaf. Sawa? Together?
Yes.
Umeandika yote? So I believe now, if you're given a procedure, whether mlifanya kitu kama mjafanya, but tumefanya practical sasa. So know that we do all these steps, all these procedures for these activities. Boiling of the leaf in the water to kill the leaf cells and stop any other chemical reaction. It's photosynthesis. The other one, boiling with the spirit. Why? Want to discard, to remove the green color. That's why water is very much green here. It means chlorophyll has moved from those leaves. Then from there, don't forget, why do we boil in a water bath? Not directly from the heat. It's because our mineral spirit is highly flammable. You can catch fire, highly flammable. Then you wash in hot water. Why? To soften it. Then the last one, what do we conclude? I will put on the water and the pour the yellowish and the brownish iodine. It will change from brownish yellow into blue-black. There was presence of starch in that leaf. Are we together? Together?
Yes.
What's our question? Yes.
Is the spirit still in you?
So, yes, it is. But it is. Pia tukimwaga hapa alafu pige kiberiti kule na maji. Sisi nada kwa shughuli. Chenye kimebaki tu kutumia tu microscope.
Yes.
Utaambia Pastor John, nataka cover slips. Cover slips. The slides.
Okay. So hiyo tunaweza fanya
An-
tukiwa
Anaweza
nazo.
anaweza nunua tu. Sinakuanga kwa katika nakaa hivi. but a shorter one. Let's go back to because kidogo sana. Na ni pamoja na the whole set of peer slides, glass slides. The whole set. The next weekend, fanya microscopy. You tumia microscope. You observe everything. You observe different cells. You observe both animal cell and plant cell. You have to make it to. So now, plant animal cell. One of you, tutamtoa kanyama hapa kwa mdomo. One of you. Si nyama. Hatuka tulimi. No. So now sometimes tunasikia wanataka vidonda kwa mdomo. Yes, just as a very tiny piece from that place now.
Nakunywa chai moto. Toma kala kapisi.
Just a small one. Then you'll always you'll see how the cell looks like. Very wonderful. So, yeah, one of you. So you just get one from here, down from there. We have another experiment.
So,
Any question? I'm here for now.
any question, you ask questions. You ask questions. It's very important. Unless you...
For my experiments here, what are we concluding? Osmosis took place. Sawa? Osmosis took place. Why? Because water moved from what was high, into the beaker, into the potato, where we have the sugar. Sawa? When you come to do microscopy, I will explain the experiment concerning osmosis, also diffusion. And then we do it here, all of us. Sawa? Because medicine are cells. Then again, we look at the similarities between animal cell and plant cell, then differences. Sawa? And also roles of diffusion and osmosis.
You're not telling me what you can say.
Sawa?
Time.
So you can find me in the next
Utakukumbusha.
next session. Next session.